Great. Well, we're half an hour ahead of schedule, so I guess I get the full hour. Is that right, sir? Great. Thanks all for coming out. Good talks to follow up on here. So I'm going to talk primarily about PacBio assembly today. I work for the federal government, so just a disclaimer that despite all the wonderful things I'll say about this technology, the US government doesn't endorse it in any way. And this is a highlight talk, which means I'm supposed to talk about a paper we published last year. The main thesis of that paper was that the majority of known bacterial and archaeal genomes could be assembled at finished quality with a single PacBio sequencing library. I have a bit of a quandary because I'll talk a little bit about that, but I'd really like to talk about some new results. So I've split it up kind of half and half. The first half will focus on bacterial assembly, and the second half will focus on larger genomes. So this work was based on some hybrid assembly work that primarily Sergey Korin, my collaborator, had worked on back in 2012. That was when PacBio first came out, and we were convinced that the error rate was too high to assemble de novo, and so we were using primarily Illumina reads to correct and assemble. And the first genome we did that with was the parakeet parrot genome. But then it turned out that hierarchical assembly strategy worked quite well as PacBio read lengths improved. And Jason Chin, who's in the room here too, had a seminal paper showing that PacBio reads themselves were sufficient to get very high quality assemblies. And so this paper in 2013, Reducing Assembly Complexity of Microbial Genomes, by Sergey Korin, myself, and others, looked at that hybrid strategy for assembling just PacBio reads and went on to show kind of how successful that is for microbial genomes. I've updated our claims from that paper to reflect the current status of PacBio chemistries. So their latest chemistry is called P5C3. And if you sequence one SMRT cell of that chemistry for a typical bacterial genome, about 90% of all of the genomes that are currently in GenBank, we predict you would be able to close using just one of those libraries. And our tests on accuracy of that show that it's about QV50 or greater when you have 100x plus coverage. And the assemblies can be generated in less than an hour and less than $1,000 total cost. That includes sequencing costs, library prep costs, computational costs. So it's really gratifying to see, as somebody that's worked on assembly for the past decade, that now we've finally kind of achieved our goal of having finished closed genomes automatically generated out of a computational process. So starting with the thesis, so I need to back up those claims a little bit. Just a quick overview, there's a lot of work going on in this field right now, and there's a lot of strategies for assembling long reads. Sergey and I just finished writing a review paper for this that'll come out on bioRxiv as a preprint in the next week or so. One chromosome, one contig is the title of that review paper, and it's going to very briefly survey all the available tools that can be used for PacBio assembly. I just had to add one to the list from the HitSeq session. This LoreDeck tool I wasn't aware of just came out, so we'll add that to the review before the preprint goes up. But today I'm primarily going to be talking about this hierarchical approach, which is what our tool, PBCR, also known as PacBio to CA, uses, Jason's HGAP assembler, and you should be highly encouraged to attend Gene Meyer's talk coming after the lunch break, where I think he'll unveil some of the inner workings of his new assembler, Dazzler, the Dresden assembler. So look for this review paper if you're interested in some of the other methods. Spades I have down here in the what I call assembly boosting section, where you already have kind of an Illumina-based Bruin graph, and you'd like to boost that and close that using long reads. So it's great to see Spades now supporting long reads as well. It's also good to give an assembly talk at the end of the session because I don't have to go into too much of the details about what assembly is, but just to drive home the point that read length and or insert length really matters when you're doing genome assembly. And so what I have here is an idealized assembly graph. So this is for k equals 50 for essentially perfect data, so no errors, infinite coverage. And this came from a paper that Carl Kingsford and others had put out a couple years ago looking at the complexity of assembling from Illumina data. So this is the E. coli K12 genome with perfect data for a k equals 50, and this is the idealized assembly graph, and each of these nodes is a contig, and any of the edges in here is some ambiguity between the contigs that can't be resolved from the short reads. So even the most simple genomes here look very nasty when you're at k equals 50. So that's kind of the short read example. If you go up to maybe Sanger-based lengths, and you have perfect data, it's possible to get it into maybe 10 or 20 contigs, but you're still not able to resolve the longest repeats in the genomes. So you're left with a handful of contigs. But even so, without any additional information, such as long-range mate pairs, the ambiguities in this graph can't be fully resolved. But then if you increase K up to, say, 7,000, that graph collapses down into a single self-adjacent node. That's because it's a circular genome, and that node is the full chromosome of the E. coli K12 genome. And so we've been calling this threshold of read length at 7,000 bases the golden threshold. And that came about via an analysis that we did for this, this assembly complexity paper, where we took all of the finished genomes that were in NCBI RefSeq, all of the microbial genomes, that is, and we did a repeat analysis on those genomes. So we aligned them to themselves, and we looked at inexact repeats at 95% identity or greater, and repeats that were 500 bases or longer. And what this plot is showing is a density of the repeat count versus the maximum repeat in the genome. And so along the x-axis at the bottom, this is the number of 500 base repeats or greater, the number of repeats in the genome that are 500 bases or greater. And you can see from the density plot, the histogram here at the top, that it's kind of just a gradually decreasing curve, so there's no real threshold in how many repeats are in some of these genomes. But if you look at the maximum repeat length, so that's the largest repeat in any one of these genomes, there is a very clearly defined threshold. So if you look at this histogram on the right, there's this huge set of peaks here between the 5 and 7 kb range, and this is the rDNA operon that exists in most bacteria and archaea. And so what this plot is showing you is that for the vast majority of genomes that we currently have finished in RefSeq, the rDNA operon is by far the largest repeat in most of these genomes in terms of frequency. So I have a couple on here. Bacillus anthracis doesn't have a whole lot of repeats, and the largest repeat it has is its rDNA operon. Yersinia pestis has a lot of repeats, but they're insertion elements, so they're about 1,000 bases in length. So it has a lot, but not a whole lot of big genomes. And then you have some of like the enteric E. coli and other that have large phage integrations, and those are the ones that have repeats that are much larger than the rDNA size. And some of those segmental duplications in bacteria can range to 100 kb, 200 kb in length. The good news is that the PacBio chemistries have been very rapidly improving over the past 4 years or so. Starting with kind of their original C1 chemistry, this is showing a length distribution of the reads that you would get. C2 is kind of the first commercially available chemistry that was widely in use. And if you look at how many reads come off of the instrument that are greater than this 7 kb magic threshold, at the time, only 16% of the reads per run were exceeding that length. So you ended up having to do a lot of runs on the instrument to collect enough of these long reads so that you could span the rDNA operons and complete the genome. But kind of year by year improvements, the next chemistry got 51% over 7 kb, and now their current P5C3 chemistry averages about 84% of the reads collected being over the 7 kb length threshold. That's assuming you do a 20 kb PacBio library prep, which has a size selection step involved. And so back to this idea of what the repeats look like in genomes, as we were looking at all of the repeat structures and all of these bacterial genomes, just kind of for convenience sake, we divided them out into these 3 complexity classes, where class 1 is essentially the non-repetitive genomes, so B. anthracis is my example here. Class 2 are the genomes that typically have been very hard to assemble because they have tons and tons of repeats, but they don't have any large repeats. And so Yersinia pestis is my example here. In this dot plot in the middle, each one of these gray dots is a small insertion element when you align the genome against itself. And so essentially across the whole length of the genome, it's nothing but these IS sequences every few kb or so. And then when you get up to the class 3 ones, these are kind of the bad boys that have both a lot of repeats and they also have very large repeats. And so on this dot plot, these red circles are kind of these phage integration sites in this E. coli that I have here that represent the hardest challenge. The key with PacBio is that the class 1 and class 2 genomes are essentially solved with the long reads. Because there's no repeats that are greater than the 7 kb threshold, PacBio sequencing isn't affected by these smaller 1 kb repeats, and so it can close it. And so if you look at how many genomes fall into these classes in GenBank, about 70% of the currently known genomes are of this class 1 type, 8% are of this class 2 type, and 23% are of this class 3 type. We're at the point now where we can confidently resolve these class 1 and class 2 genomes, and we can resolve maybe about half of these class 3 genomes depending on what the repeat structure looks like. So that's where we get our estimate about 90% of the known genomes can be closed using PacBio. This comes with the caveat that this is the genomes that are currently in RefSeq, and so it kind of is selecting for the easier to finish genomes. And so I doubt this is a true reflection of what the repeat complexity looks like in nature, but it's all we have to go on for now. So kind of the summary and the defense of that thesis that I first presented was with some restrictions applying, microbial sequencing can be solved with long reads, that the majority of the available genomes can be closed. When we did a validation analysis on those against the available reference genomes, we showed that the QV accuracy was about 50 or 60, so that's 99.49's worth of consensus accuracy, and that's largely due to the Quiver tool that PacBio developed that can use the PacBio chemistries to make very accurate, can use knowledge of the error models of the PacBio chemistries to make very accurate consensus calls. And so after we looked at that, and we've been pretty happy in using the PacBio as kind of our default microbial sequencing machine, we're moving on to the next challenge, which is large genomes. So can we extend the success that we've had for the microbes to the insects, the plants, and the mammals? So that's what the second half of my talk will be about, and that I'd like to focus on. The sequencing problem isn't completely solved for microbes. There's still a lot of engineering challenges in making the assemblers more robust and less likely to fail, and more robust in the corner cases. Another key element is graph-based assemblies and formats. And so one thing we've noticed is that a lot of these microbial cultures have very dynamic chromosomes. And so you'll have gaps in your assembly that might not be caused by repeats, but it's caused because there's a inverted element that's flopping between two orientations in the genome. And it's hard to put that into a linear structure and report that as a FASTA file. And so having some graph-based representation of the assembly would be useful. And then diploid, polyploid, metagenomes, all of these examples require graph-based formats. And so I think that's certainly the future of assembly, is ways to report that graph back to the user rather than just a linear FASTA file. Also, if we have closed bacterial genomes, we might be starting to move out of the era of read mapping. And so we need tools that can work on these whole genomes. You know, this was a hot area of development back in the early 2000s, but the short read era has kind of moved everything to mapping. We have to go back now a little bit and start looking at these tools again for doing whole genome analysis, and so my group has started to work on that again. Efficient algorithms for assembly are critical. So the first versions of HGAP and PBCR that were used to close microbial genomes would take a day to assemble a microbe. And so trying to scale that up to larger genomes wasn't feasible at first. And so we've been working very hard on improving the efficiency of these algorithms, and that's what I'll be talking about for the majority of my talk. So just a quick note on tools for assembled genomes. This is a preprint that should be out this week. We were trying to have it out for ISMB, but we didn't make that deadline. But this is by Todd Triangan, my collaborator, on rapid core genome alignment and visualization for thousands of microbial genomes. So assuming one day we have a situation where we have thousands of closed microbial genomes, we'll have the tools ready for doing that whole genome alignment and analysis, mostly for outbreak attribution and analysis of highly related strains. And so this is showing the whole genome alignment of about 1,000 E. coli genomes, giving you their whole genome SNP tree, and allowing you to zoom in and out of the different SNPs and phyletic patterns that are in that genome. So I encourage you to keep an eye out for that if you're into microbial genomes. But on to large genomes, which is the most interesting part of this. So PacBio sequenced the Drosophila Melanogaster reference strain. So this is the exact strains, the exact same subline that Celera Genomics sequenced back in 2001. It's about 160 megabase genome. PacBio sequenced it at very high coverage, 90x, which I estimate is about a $10,000 to $20,000 sequencing cost. In collaboration with PacBio and Casey Bergman at University of Manchester, we assembled this. And because they had collected so much data and the new chemistries are so great, we were able to assemble the genome by just selecting the 25x longest reads. And when you do that, you end up making an assembly entirely out of reads that are 17 KB or longer. And so, I mean, this is every assembler's dream. When you're given 25x coverage of the genome and every single read in that data set is longer than 17 KB, it certainly simplifies things. So running this through PBCR, which is implemented as a module of Celera assembler, we got very impressive assembly stats out of this. And so we had a 164 megabase assembly, which was roughly the size we would expect for the genome. The maximum contig was 26 million base pairs, and the N50 size was 21 million base pairs. The important things to note is that this wasn't just an exercise in showing what we can do, but it's actually scientifically valuable data because there's gaps that this assembly closes in the Melanogaster reference sequence, which is by far one of the most well-studied reference sequences and has been undergoing manual closure for the past decade. And so there's gaps in there that, back sequencing, phosmid sequencing, Illumina, Sanger, all of the available technologies have been thrown at this genome over the years, and there's still persistent gaps that this assembly closes automatically. And then here's showing kind of the autosomal chromosome arms and how many contigs came out of the assembler, and it's at most 6, in most of the other cases 1 to 3. And so there's chromosome arms in this assembly that are assembled from telomere to centromere into a single piece. The nice thing, because this is the same subline that Celera is sequenced, we have this reference genome that we can use to assess the accuracy, and the structural accuracy out of the Celera assembler is largely correct. This chromosome arm 3L I mentioned actually has kind of the full telomeric sequence on one end, and it goes all the way across the autosomal portion into about 2 or 3 megabases of centromeric sequence. And so it's from telomere to centromere, and attached to that are large chunks of the heterochromatin. The mess down here at the bottom is the X and Y chromosomes and a bunch of the heterochromatin that didn't assemble very well. The large chunks, the large continuous chunks in this dot plot are the autosomal portions. So with the longer reads and the longer contigs, you also start to capture longer repeats. And so this was an example, I think, that Jason Chin had dug up that's showing some of the repeat structure that's on the Y chromosome of Melanogaster. And so in the middle of this dot plot here, it's showing this very complex kind of tandem array that spans 300 kilobases on the Y chromosome. This kind of stuff was never assembled before. And this came out into a single contained contig from the PacBio assembly. And there's actually genes interspersed in this mess, if you can believe it. It looks like it might be uninteresting, but it does contain genes, and it is important for the function of the organism. Particular improvements in the rest of the assembly, the Y chromosome is essentially uncharacterized in Drosophila. So the previous coverage of that chromosome was maybe 2% of the chromosome length. Our collaborators estimate that now it's maybe up to about 40%, 50% of the chromosome length is characterized by this assembly. We have telomeric and centromeric gaps that are closed by the assembly, and the transposable element resolution looks excellent. So there's some known TE families that are fully resolved in the assembly. So great results, but it all came at a huge computational cost. And so we originally ran this using the methods that we had developed for bacterial assembly and that PacBio had developed for bacterial assembly. Primarily, we were using Blazor for overlapping, and we were using the Celera assembler for contigging. This ran on my cluster for a month straight, which comprised about 630,000 CPU hours. And so when we first released this result, there was a lot of kind of pushback that, well, this is a great result, but who has that much compute power to run this on their own? And we agree wholeheartedly with that. And so we were definitely in need of a faster solution. And so for the past year or so, we've been working diligently on that. Key to note here is that the overlapping runtime is comprising 608,000. The total runtime for the assembly was 630,000. And so 98% of the time in the assembly was spent just in the overlapping step. And this isn't a bash on Blazor at all. I mean, Blazor is kind of defined and designed as a read mapper. There's efficiency techniques that can be used for assembly overlapping that it doesn't include. And so it's a great mapper and it's very effective at that, but we need something that's solely dedicated and designed for overlapping. And so Sergey Korin, who's my collaborator at NBAC, turned to his close friend who was a postdoc at the time at University of Maryland, Konstantin Berlin. And Konstantin comes from kind of the protein realm and the scientific computing realm, so he was aware of a lot of algorithms in the space that might be useful to us, but he wasn't in kind of the assembly and sequence alignment mindset that Sergey and I have been in for the past 10 years. And so we described the problem to him and kind of immediately said, well, this is what you guys should be using. And he came to us with this idea of min-hash, which is kind of also known informally as the Alta Vista algorithm. It was invented in '98 by Andre Broder, and it was, when he was at Alta Vista, it was designed as a method for detecting duplicate web pages. So if we think of our sequencing reads as web pages and our question for overlapping is to find ones that are similar, there's some analogies here to finding similarities between two documents. And what this min-hash process does, it's a quick estimator of a Jacquard score, a Jacquard similarity. So a quick overview of what a Jacquard score is. We're all familiar with the identity scores. You Smith-Waterman, align two sequences that I have here, and you get 50% identity, just meaning 50% of the bases are shared. A Jacquard score, in contrast, decomposes both sequences into their K-Mer sets, and it's really a set similarity score. And so it's saying the intersection of all of the K-Mers between those two sequences divided by the union of all of the K-Mers in those sets, and it's essentially just measuring the fraction of the K-Mers that are shared between those two sequences. And it has a couple of nice properties. It's position independent, so it's kind of alignment-free method. You can just look at the K-Mer sets and not worry about alignment, but it's also strongly correlated with identity, obviously, because the K-Mers that comprise the sequence, if the two sequences are 100% identical, all of the K-Mers will be shared and you'll have a perfect Jacquard score. So it's relatively quick to compute, but still you have to look at all of the K-Mers and all of the reads, and that's a bit of a slow process. And so min-hash is designed to be an estimator of the true Jacquard score. Prelude to what min-hash is just to talk about K-Mer hashing. So we don't want to actually compare sequences of K-Mers. We want to just compare something much faster, like an integer. And so very typically, K-Mers will be hashed into K-Mer fingerprints, which are essentially binary representations of the MER, use of a hash function. And then here I have the MER cat going into a hash function, coming out as a K-Mer fingerprint, and then this is its decimal representation. The key idea in min-hash is that there's more than one way to do this. You don't need a single hash function. So if we run this through, currently we're using the murmur3 hash function. We run it through, we get a K-Mer fingerprint. You can take that K-Mer fingerprint then and use it as a seed for a pseudo-random number generator. And what this does is randomize in a deterministic way your fingerprint into many different fingerprints. The next slide will explain why this is useful, but just to show that this same K-Mer can be hashed into these 4 different decimal representations. So this is just an example. We use 32-bit fingerprints in MHAP, which is the algorithm I'm describing here. Here I'm just using 6 bits for an example. So what a min-hash sketch does in the case of DNA sequencing, I mean, it should be obvious how this would apply to a document where you have English or any type of words. You take your DNA sequence and you decompose it into its set of K-Mers. So here we're just using K-Mers of size 3 for an example. You take the first K-Mer and you run it through our 4 hash functions, which essentially is running it through the murmur3 hash function and then using an XOR shift pseudo-random number generator to make different versions of this hash value. So here I've got cat and I've got its 4 hash fingerprints. Then you do that and repeat that process for all of the K-Mers that are in your original string, and you end up with a list of all of the MERs that were in that string and these 4 different hash functions outputting their fingerprints. And so min-hash is a dimensionality reduction formula, which is taking this 20 base string and trying to reduce it into a smaller size. And what you get from this process is instead of keeping all of the fingerprints from each of the hash functions for each of the MERs, you just remember the minimum valued fingerprint that came out of the hash function. And so for hash 1, I'm going to remember 5, and we're going to call this the min-MER. It was the K-Mer that generated the smallest valued fingerprint for this K of ACC. For hash 2, we have 1. For hash 3, we have 2. For hash 4, we have 14. And so these 4 values highlighted in red are the 4 min-MERs that are going to be remembered by this min-hash sketch. And so the sketch size can be adjusted, but here I'm just using a hash sketch size of 4. And so the min-MER sketch for this string of length 20 is just these 4 integers. And the nice quality of this is it's called a locality-sensitive hashing function. And that means that if you get this min-MER sketch of these 4 values and you run a similar sequence through the same process, if the 2 original sequences were similar, there's a very high probability that their resulting sketches are going to be similar. And that's a result of, if you have an ACC in your string A, and you have an ACC in your string B, it's very likely that those 2 will result in the minimum value out of the same hash function. So to use this for alignment, you take your 2 sequences that you're testing. It's essentially an overlap filter. We want to test if these 2 sequences overlap one another. You take sequence A, you decompose it into its K-Mers, you run it through your hash functions, you generate its sketch. You take sequence B, you decompose it, you run it through the hash functions, each time remembering the minimum value for each hash, and then you build its sketch. And here I'm showing that we also index where these min-MERs came from. So for hash function 1, 5 was the min-MER. I can trace that back and know that that came from ACC. And the result is these 2 sketches. Now the question is you need to compare the similarity of the 2 sketches to determine the similarity of the 2 original sequences. And the key fact to all of this is that the probability that 2 of these elements are going to be equal between the 2 sketches. So for element 1 to equal element 1 in A and B, the probability is exactly equal to the Jacquard similarity of the 2 original sequences. Okay, and so what you end up with as you start sampling more and more how many of these hash functions have equal values, the more and more that you have similar values, you build up a sketch of length 4 here, and I've got 2 min-MERs that match, and that's giving me a Jacquard estimate of 0.5 for these two sequences. And so because the probability that these elements are going to be equivalent is equal to the Jacquard similarity, it ends up being kind of a sampling process. And as you extend your sketch size larger and larger, you start to reduce the error that's in that estimator. And so you can mathematically predict what the appropriate sketch size will be to bound your error on the Jacquard distance. And so then knowing an estimate of the Jacquard distance between those two sequences, you can use an index to go back and find their min-mers, and that's how you do the overlapping. And so I knew that I had two matching min-mers for these two sketches, 5 and 1. I trace 5 back to ACC, I trace 1 back to CCG, and then in the original sequences, you know that this is a seed for that alignment. And you can use those seeds to determine the offset between two sequences, and boom, you have your overlapping information without ever having to do a Smith-Waterman alignment. And so the proof in the pudding for all of this is the performance that comes out the other end. So I was initially skeptical of this idea when Sergey and Konstantin brought it to me. I'm kind of the old school that likes to see the full alignment, you know, the full Smith-Waterman, have everything be very thorough. I was a little uncomfortable with this idea of a sketch, but when they brought the results to me, I was fully convinced. And so just the caveat that those two guys are the ones that did all of the work here, and I'm just here to present it for them. And so when we first ran the Drosophila melanogaster assembly back in December of 2013, I said this took 630,000 CPU hours to make that Drosophila assembly. When we repeated this process using the map, the min-hash alignment process, overlapper, that was the only module we subbed out in addition to a new correction consensus module developed by Jason Chin, and it reduced the compute time from 630,000 down to 1,000 CPU hours. Most of that savings obviously was in the overlapping step, so the speed up was 674-fold speed up just for the overlapping alone. And key to all of this is that not only did we speed it up, the assembly actually got a little bit better because the map process was more sensitive in finding overlaps in this genome than Blazor was. And so swapping out only the overlapper and the correction code, we ended up with the same number of contigs, but the N50 length went up from 15 megabases to 20 megabases. And so it sped up. The assembly got more continuous, and when you align back to the reference, it's still a correct assembly. Our collaborator, Casey Bergman, took our Solera assembler process for generating these assemblies, and Jason bundled up a recipe for him so he could try it out on Amazon Web Services himself, and he was able to reproduce our exact assembly using these methods on Amazon Web Services for less than $300. For people that care, the parameters that were used for that overlapping was a K-Mer size of 16, a sketch size of about 500, and a min-match of 3, meaning of that sketch of 512 min-mers, we required a minimum of 3 of them to be in common for it to pass the filter. So we now started to extend this method to chromosome scale assembly of large genomes. PacBio graciously generated and deposited all of these genomes on the left here. All of that data is currently going to SRA. It's also available on AWS. If you go to PacBio's website, you can download it and play with it however you like. That kind of triggered the development of all of these algorithms because we love to play with data. So they had E. coli, Cerevisiae, Arabidopsis, Drosophila, and human, and an idea of the coverage. They're all very high coverage, very modern chemistry results. And so in testing MAP, we've gone through and assembled each one of these genomes and assessed their accuracy and assessed their continuity. The results look fantastic for all of them. And in addition to Drosophila, this was all kind of a prelude to helping our collaborators at the USDA who want to assemble the goat genome. And these guys, I think, get the award for the most use of their PacBio instrument. They sequenced almost 500 cells of PacBio of this goat, generating about 71x coverage. And so they had told us about this project back in the fall, and we took a very deep breath because this was before we had MAP, and we had no idea how we were going to assemble this because our current algorithms were too slow. But now we were able to assemble this goat genome in about a week. And the resulting contigs of this goat genome, all of these genomes on the left here are kind of special in some ways. So Drosophila melanogaster is very inbred. This human is a hydatiform mole haploid cell line. Arabidopsis and yeast and E. coli, they're all kind of like haploid, very simple organisms. This goat was the first kind of true diploid organism that we tried to sequence. It's about a 2.6 gigabase genome. And the result out of this min-hash enabled assembly process is a contig N50 of about 1.5 megabase pairs. And so this goat genome had been assembled previously with BGI using Illumina, and they had long-range mates, they had short-range pairs, and the result of that assembly, the scaffolds that came out of that assembly are smaller than the contigs that came out of our PacBio assembly. So this has our collaborators very excited. So MAP is available now. We haven't released the preprint on it yet. That'll come by the end of the month, but the software is up there and we have people that are actively using it. If you don't want to remember this long URL here, you can just Google PBCR for PacBio corrected reads map, and that should send you to the wiki that has instructions on running it. PacBio also did a webinar recently that Sergey gave a live interactive demo, so you can go watch that on PacBio's website if you'd like instructions for how to run it. And we're excited by the end of this month to release a preprint on efficient assembly of large genomes using single molecule sequencing and locality sensitive hashing. And Konstantin was the lead on developing the MAP algorithm. Sergey has been running all the assemblers and doing comparative analysis. Jason wrote the consensus module that we're utilizing and is of HGAP fame. James Drake and Jane Landelin, also at PacBio. Casey Bergman, who's our resident Drosophila expert, and myself. And then just finally a call out that everybody should go see Gene Meyer's keynote after the lunch break. You know, he's one of the godfathers of assembly, and I'm sure he'll have great things to reveal for his Dazzler assembler. And sorry to those that came in. We started the session half an hour early, so feel free to come up and catch me if you want me to go through the presentation again. And I'm happy to take any questions. Thank you.
Hi, that was a great talk. With the Drosophila resequencing with PacBio, was that from the same DNA, genomic DNA from when they sequenced the fly a decade ago?
Right,
or
so
was it a new fly from the strain?
Yeah, so it's not the same DNA as in they didn't grind up a bunch of DNA when they started that project and keep it in the freezer for 10 years. But they have maintained the same subline of insects over the past 10 years. They've been continually using for the Drosophila reference project when they need new material. And so it's from the same strain, the same subline, essentially the same lineage of flies.
Right, so I'm curious if you guys have looked to see if in that strain, you know, comparing it back to the reference genome, if there's a lot of changes, how stable is that genome?
Right, and so it's hard to tell too because there's generally not enough material in a single fly to do sequencing of any sort. And so the original genome project itself, I think, is an amalgam of multiple insects. We're doing a mosquito project, and it's the same thing. You need to collect like 200 mosquitoes and grind them all up to get your DNA. And so the original reference assembly itself is an assembly of a population of insects. Oh, it's just it's a very inbred line, and so it helps for assembly. And so we haven't looked to see if there's any population level differences between them because that's one of the complications, but it's a cool idea.
Okay, thanks.
Yep.
Adam, I really enjoyed your talk. Just with respect to the MAP assembler,
yeah.
How do you think this would work in the context of high levels of heterozygosity? So we're actually making a new, I'm working with Mike Lynch to do a new Daphnia pulex reference. That's not inbred. It's got high levels, high effective population sizes. So I'm trying to convince them about this technology, but could you speak to that?
Yeah, the heterozygosity issue and the diploid and polyploid issue is a hot button topic right now. And so MAP as an overlapper would do a good job of detecting the overlaps that then you would want to use in the assembly process. What we're currently missing is the contigging step that takes those overlaps and turns it into a graphical representation of the genome. So Jason Chin from PacBio has been working on this Falcon assembler, which builds a string graph and then tries to walk different paths through that. But still, his algorithm outputs a mosaic of the different haplotypes, so there's switching errors all throughout it. But the idea is to get some large-scale structure of the genome. And so the information, without a doubt, is in the reads, and I think you could do a very good job of phasing it. It's just the algorithms haven't quite caught up there to exploit that data that's in the long reads. And so we're interested in, for the GOAT project as well, we're doing a high C sequencing project to get this chromatin interaction level data that we can use for long range scaffolding. There was papers last fall for using that for both scaffolding and haplotype phasing. And so we're excited to look at the combination of PacBio and this high C Illumina sequencing to do phasing and full assembly of the chromosomes, but there's no methods that do that right now. And so we'll have to wait a couple of years for people to develop that.
Thank you.
Thank you, Adam, for a great talk. One question, not very technical. So what is the minimum coverage of the PacBio so that you can assemble? I mean, for mammalian genomes, because we study with human.
So it depends if you're doing a hybrid or just all PacBio. So when we did the the parakeet genome. We only had 4x of PacBio sequencing and showed that it could be used to improve 15x of 454, which
we
is
would
what we
like
had
to
at the time.
study the line 1
Yeah.
repeat, so it should be.
So if you're using just PacBio, this original slide I had here is kind of showing the minimal coverage. And so Celera assembler developers have tried to just run Celera assembler on low coverage data like 10 or 20x of PacBio with limited results. This hierarchical method that I propose now to do this self-correction and assemble these very long contigs requires about a minimum of 40 or 50x coverage. But there are these alternative approaches. So Mike Schatz's group has this EC Tools program which pre-assembles Illumina data, uses that for correction. So there, if you have maybe 50x of Illumina, you can get away with 10 or 20x of PacBio. But for the really low coverage options, you're left with these assembly boosting techniques, which is you build an Illumina assembly first, and then you layer on your low coverage PacBio to fill the gaps and improve the assembly. So if you're just starting with PacBio and you want to assemble PacBio only, the minimum coverage is about 40x, I would say, to get a decent assembly right now.
But this is great. Another question, what is the cost to generate 50x coverage for human genome? Do you know?
So for human genome, I have not made that calculation yet, but I
For
assume
example,
it's
goat.
not cheap. So my estimate for Drosophila was about maybe $15,000 if you just look at what core sequences are charging per smart cell.
And for the goat one that you sequence?
I don't know, I didn't see it. That was the USDA, so you can ask
Okay.
them. Apparently the goat is very important to them. I'll just go to the cells. And so if you look at the Drosophila, so it was about maybe $15,000 for 42 cells. So do the math and you can see that it was probably over $100,000 for them to do the goat genome, and that would be a rough
Quite
price for the
expensive,
human
huh?
genome as well.
Okay.
So it's also important to keep that in context that the Melanogaster reference genome project has been going on for 10 years doing back sequence, paired end backs, so on and so on. And there's been hundreds of thousands of dollars spent in those manual closure projects. And so what we're trying to show is that now in 1 month's time, you can get a similar result for roughly a similar cost without that 10-year worth of
Okay.
effort.
Thank you, Adam.
Yeah.
What do you think about the future of PacBio reads for metagenomes?
So we've looked a little bit at metagenomes. It's a little tricky in some regards because our correction approaches are aligning reads to correct out the errors. And when you're in metagenomes, you don't have this haploid or diploid assumption, and so that makes it harder to be confident that you're accurately correcting the reads. The other issue is the throughput of the instrument. So, compared to Illumina, it's not nearly as high of a throughput instrument as some of the others, and so that limits what you can look at. So for very low complexity metagenomes where you have maybe 10 different species or something like that, I know people looking at kind of acid mine drainage pools have used PacBio for this. I think for that it could be exploited pretty well. For the very high diversity metagenomes like human gut and soil and things like that, I think you have to wait till the throughput gets a little bit better. But in a metagenomic scenario, it's certainly very intriguing to have these long reads, maybe not for assembly to begin with, but just for mapping to the reference databases to get a very accurate call of what's in the metagenome.
Thank you.
A quick, more technical question. In
Yeah.
terms of you had a chance maybe to see different machines working. Now, obviously, PacBio has this machine very well working. The goat, it wasn't sequenced in for the PacBio machine, but not in PacBio. Was there a difference in the reads? Were they shorter? Were the libraries not as good?
Right, so there is some variability in different groups in using their PacBio instruments. So all of the genomes on the left here were generated in-house by PacBio. The goat on the right was generated by the USDA in their facility in Nebraska. And I like to collaborate with this guy, particularly Tim Smith at the USDA, because in my experience, he's one of the best at running his PacBio instrument, and so there very much is variability. In our center, we often have times getting good libraries, good long reads, good loading on the instrument, and some people seem to have the magic touch. And so there is variability across the different centers in collecting long reads.